Hi, I'm David, and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Question 1 talks about smooth muscle. 1a. Explain how smooth muscle cells in the wall of bronchioles contribute to the function of these airways. Smooth muscle is an involuntary muscle. It contracts and relaxes automatically without our conscious thought. It can change the diameter of bronchioles. Its contraction decreases the diameter of the lumen, while its relaxation increases the diameter of the lumen. This regulates the airflow. A reduction of the lumen diameter restricts the airflow. There is a mark for AVP. You can state one condition when they need to contract to reduce the airflow. For example, in response to allergens in the air. Note that you should not use the terms vasoconstriction and vasodilation, which are used when describing changes in lumen diameter for blood vessels. The right terms are bronchoconstrictions and bronchodilation. Figure 1.1 shows a smooth muscle cell. A stage micrometer is used to calibrate the eyepiece reticule. It is a microscope slide with a precisely marked scale of non-axial length. To know if the student can see the cell, we have to change the values to the same unit. We can convert millimeters to micrometers by multiplying the value by 1000. So, the smallest size the student can see is 200 micrometers. With that said, the cell can be seen as it is larger than the limit. Figure 1.2 shows the smooth muscle tissue. A capillary with red blood cells is visible in the photomicrograph. 1. Outline the features that help to identify the blood vessel as a capillary. For this type of question, ensure that you answer based on the photomicrograph provided. First, the wall is one cell thick. It only comprises a single layer of endothelial cells. A capillary is the only type of blood vessel that has a one cell thick wall. Due to the small lumen, red blood cells are transported in a single file. Other blood vessels, like arteries and veins, have a larger lumen. Red blood cells do not pass through them one by one. Next, we can see the red blood cells fit tightly into the lumen. This means their diameters are approximately the same size as the capillary lumen. Other blood vessels would have lumens that are way larger than this. The diameter of the capillary is narrower than that of a smooth muscle cell. This is because a capillary is the tiniest blood vessel we have. There is a mark for AVP. You can discuss the structures that should be present in other blood vessels but are not visible in this photo. You can also discuss how red blood cells change their shape slightly to pass through the narrow capillary. 2. Explain how the structure of capillary is related to its function in smooth muscle. The first mark is given when you state the function of a capillary by naming any substance that it carries to the muscle or away from it. Some examples include oxygen and glucose for respiration, and amino acids for protein synthesis, the removal of waste products such as carbon dioxide from respiration, and lactic acid from anaerobic respiration. Then, relate its structure to this function. A capillary has a thin wall. It is only one cell thick. This creates a short diffusion distance, increasing the rate. The wall has many pores or fenestrations. This allows substances to move in or out without passing through the cell, leading to a more efficient passage. Endothelial pores also allow the passage of phagocytes such as monocytes and neutrophils. They can move to the site of infection and perform phagocytosis. Capillaries have a narrow diameter. They can penetrate the tissue and reach all cells, allowing material exchange to occur between the cells and the blood. Their small diameter also slows down the rates of blood flow, allowing for a longer time for the exchange to occur. The last mark is given if you relate its structure to red blood cells and explain how this benefits their functions. For example, you can discuss how red blood cells must pass through slowly, one by one, leading to a longer time for exchange to occur and a shorter diffusion distance. D talks about a protein with two forms and the gene that codes for it. 1 wants you to explain what is meant by gene mutation. Gene mutation refers to a change in the sequence of base pairs in a DNA molecule. You must use the word sequence here. Do not just say that there is a change in DNA. This change in sequence causes a change in the codons. Different tRNA with different anticodons will bring different amino acids to the ribosomes. It results in the production of an altered polypeptide. In other words, the amino acid sequence or primary structure of the polypeptide it codes for is altered. D2 wants you to suggest how different forms of proteins can be produced from the same primary transcript. A primary transcript is the initial RNA molecule produced directly from a DNA template strand during transcription. It contains both coding regions, exons, and non-coding regions, introns. It must undergo post-transcriptional modification to become the mature mRNA. One of the processes is called RNA splicing. Introns are removed, exons are joined together. The exons can be joined in different sequences or combinations, forming mature mRNA with different nucleotide sequences. This means that a gene can be used to produce polypeptides with different primary structures. This is known as alternative splicing. You can also use this name in the first point. 3. Suggest how the two different forms of cadosmons can still have similar functions, even though they have a different primary structure. They may have similar tertiary structures, resulting in similar shapes of their binding sites. The differences are found in the other regions. The question says that H cadosmon has a repeating sequence in the middle of the amino acid chain that is not present in L cadosmons. The removed amino acids in L cadosmons are probably not structural amino acids and are not part of the binding site. So, their absence does not change the function of the protein. Besides, proteins with different primary structures may have very similar R-group interactions, resulting in a similar 3D shape despite not being identical. There is a mark for AVP. Maybe they do have some differences in some of the binding sites, but there are still some very similar ones. Or maybe the difference is the number of binding sites they have, not the types. 2a wants you to name a few cell structures according to their functions. The nucleolus is the site of ribosome manufacturing. Inside the nucleolus, the genes of ribosomal RNA are transcribed, processed, and then combined with ribosomal proteins to form the small and large ribosomal subunits. SER is the site of lipid synthesis. It contains the enzymes needed for the process. Centrioles organize microtubules of the cell cytoskeleton. The cytoskeleton is a dynamic network of protein filaments within a cell cytoplasm that provides shape, structural support, and enables cell movement. A2 wants you to draw and label the cell structures described in Table 2.1. Nucleolus appears to be roughly circular within the nucleus. The smooth ER is a tubular structure with no ribosomes. Centrioles resemble two cylinders lying at right angles to each other. They are usually found near the nucleus. B talks about alveolar macrophages. 1. Outline the sequence of events that lead to the formation of the phagocytic vacuole after detection of the bacterial cells. The process is called endocytosis or phagocytosis. You can also get a mark by describing the process. The cell surface membrane of the phagocyte surrounds the bacterial cell and engulfs it. Any detail of events that occur before or after the engulfing will give you the second mark. For example, you can talk about chemotaxis. It is the migration of phagocytes toward a source of chemical attractants, such as those released by pathogens. This process guides them to the site of infection or injury. You can also talk about the binding of the antigen on the bacterial cell to the receptor on the phagocyte. Opsonization may occur as well. It is a process where antibodies bind to the antigens on pathogen, tagging it to make it more recognizable by phagocytes. Parts of the membrane fuse together after the engulfment to form the phagocytic vacuole. The cell structures that fuse with the phagocytic vacuole and release hydrolytic enzymes are the lysosomes. They contain digestive enzymes that can break down the components in bacteria, such as proteins, nucleic acids, carbohydrates, and lipids. C. Suggest and explain the advantages of using the BCG vaccine for the prevention and control of TB. The weakened pathogens can replicate in our bodies because it is a live vaccine. This means that the number of cells can increase after the administration. When there are more cells, there will be a higher level of non-self antigen. A larger quantity of antigens may trigger a stronger primary immune response, leading to the production of memory cells. They provide immunological memory. So, vaccination is a type of artificial active immunity. If the cells can replicate, reintroduction of the same antigens is not required. So, boosters are not needed to build sufficient immunity. The vaccine does not cause TB because the cells are weakened or attenuated, making them unable to cause the disease. The cells have been modified to be non-pathogenic in humans. The question also says that the vaccine is freeze-dried. It is the preferred method for stabilizing live attenuated vaccines. This type of vaccine has excellent stability. Therefore, it is easy to transport and store, enabling it to be distributed to various locations. There are 2 marks for AVP. You can discuss the effectiveness and how an effective vaccine can lead to herd immunity more easily. Question 3 talks about lysozyme, an enzyme that catalyzes the hydrolysis of glycosidic bonds present in peptidoglycan molecules. A. Draw labeled and annotated diagrams in the space provided to show how the log and key hypothesis works. First, draw the enzyme and the substrate. Label the active site and make an annotation about its complementary shape with the substrate. In this hypothesis, the enzyme active site is the log, and the substrate is the key. Their binding forms the ES complex. After the reaction, products are formed. A water molecule is used since this is a hydrolysis reaction. B wants you to compare lysozyme and penicillin. Both kill bacteria by causing the cell to lyse or burst. They act on the peptidoglycan cell wall. When the cell wall is weakened, it can no longer withstand turgor pressure. The cell bursts when water enters by osmosis. However, lysozyme is an enzyme, but penicillin is an antibiotic that acts as an enzyme inhibitor. An antibiotic is a chemical substance that kills or inhibits the growth of bacteria. With that said, their mode of action is not the same. Lysozymes hydrolyzes glycosidic bonds in the peptidoglycan molecules, but penicillin inhibits the enzyme in bacteria called transpeptidase. It is an enzyme that catalyzes the cross-linking of peptidoglycan in the cell wall. By inhibiting the enzyme, penicillin prevents the formation of the cross-linkages, thus weakening the cell wall. This process only occurs when the cell is growing and synthesizing new cell walls. So, penicillin is only effective against growing cells. Lysozyme is effective at all stages. There is a mark for AVP. You can use your general knowledge about enzymes to state some features of lysozyme that are not likely to be true for an antibiotic. 4a wants you to describe the structure of a cellulose molecule. Stating the monomers will give you the first mark. This is a mandatory point. All the other points can only give you a maximum of 2 marks. Condensation reactions occur between the monomers to join them together. The bond formed is a 1,4 glycosidic bond. The hydroxy groups on carbon 1 and 4 in a beta-glucose are not found on the same plane. So, each glucose molecule must rotate by 180 degrees compared to the adjacent molecule for the bond to form. The repeated condensation reactions cause the formation of a linear chain. It has no branches. The question asks about a cellulose molecule, so you don't have to talk about the formation of microfibrils and macrofibrils. B provides some information on the enzyme nitrate reductase. 1. State one benefit to leaf cells of an increase in mRNA molecules of gene NR after the addition of nitrate. The command word state indicates that you only need to provide a brief and precise answer. Detailed elaboration is not required. To produce an enzyme, cells need to carry out transcription and translation. When more mRNA is produced, the rate of translation of the enzyme increases. So, there will be an increase in the enzyme concentration. This ensures greater metabolic efficiency, as the enzyme is only produced when needed. This is a strategy to save energy and materials in cells. There is a mark for AVP. You can discuss how the concentration of the substrate regulates the rate of product formation and the use of nitrite in cells. 2 talks about an experiment that determines nitrite concentration by observing a color change. You are asked to suggest why using a calorimeter can improve this method. A calorimeter is a device that measures and compares the color of a sample. We can then quantify the concentration of a substance. Light is allowed to pass through a cuvette that contains the solution. The amount of light detected at the other end is expressed as absorbance. A solution with a greater intensity would absorb more light, resulting in a higher absorbance value. This provides quantitative results, as the result is numerical form. This is more accurate than color observations, as the results are not subjective. It can detect the slightest change in color intensity, which may not be visible to the naked eye. The different shades of color are correlated to different concentrations of nitrites, indicating different rates of nitrate uptake. Color observations by the eyes would not allow us to determine the rate accurately. We can use the result to produce a calibration curve. It is a graph of absorbance value against concentration of the substance. There is a mark for AVP. You can discuss how it can detect very low concentrations even if the sample is only faintly colored. C1. Outline and explain the sequence of events that occur which allows amino acids to be transported from the apoplast into the cytoplasm of a companion cell. Protons are actively transported out of the companion cell. They are pumped into the cell wall or apoplast. A proton gradient builds up in the cell wall. Protons then move back into the companion cell by facilitated diffusion, down the concentration gradient. They move through a cotransporter. Amino acids are cotransported with protons. A cotransporter is a membrane transport protein that couples the movement of 2 different substances across a cell membrane. Amino acids are transported against their concentration gradient. This process is called secondary active transport because the movement itself does not require ATP directly. However, energy is needed initially to create a proton gradient. 2. Suggest why amino acid transporters are not needed to move amino acids from the companion cell into a phloem sieve tube element. There are many plasmodesmata between the companion cell and the sieve tube element. Plasmodesmata are microscopic channels that perforate plant cell walls, creating direct cytoplasmic connections between adjacent cells to facilitate the exchange of molecules. So, amino acids only have to diffuse across plasmodesmata down the concentration gradient. They do not have to pass through the cell surface membrane. This shows an investigation measuring nitrate uptake in maize plants over time with different concentrations of nitrate solution given. You are asked to describe the differences in the rates of nitrate uptake for 5.0 and 0.2 mmol per dm cube nitrate solution between 2 and 5 hours. Since the question asks you to compare the rate, focus on the gradient of the graphs, not just the uptake at any one point. The rate is mostly constant for 0.2, but it changes over time for 5. The steeper gradient shows us that 5 has an increasing rate. In a descriptive question, the use of data will give you a mark. You can calculate the rates to support the second point. E shows us some experiments to investigate how nitrate uptake may be affected by different factors. You are asked to discuss how nitrate ions are taken up and the factors that affect this process. Let's compare each experiment with the control to determine the effects. 1 shows that a lack of oxygen reduces the uptake. 2 shows us that a low temperature decreases the uptake. 3 indicates that a lower rate of protein synthesis causes a decrease. 4 shows that the uptake only decreases slightly when bacteria are killed. Experiment 1 is a strong indicator that active transport is involved in the uptake. It requires ATP. Without oxygen, aerobic respiration does not occur. Therefore, ATP production is low, and less energy is available for the process. You can get a mark by comparing any of the experiments with the control. You may use any points I have stated just now. Then, quote comparative data to support your second point. You are free to choose from any of the 4, but ensure that your second and third points refer to the same comparison. The discussion of the link between oxygen and active transport will give you 2 marks. However, the uptake is not completely zero when there is no oxygen. This indicates that some ions may enter the cell through facilitated diffusion. Lower temperatures lead to a lower rate of respiration, as it is an enzymatic reaction. It also lowers the action of the carrier protein in transporting the ions as kinetic energy decreases. Protein synthesis affects the process by reducing the number of protein needed for active transport and ATP production. For example, carrier proteins and the enzymes needed for ATP production. Bacteria only decrease the uptake slightly. This may indicate that bacteria found near the roots can increase the uptake, but not in a significant amount. 5a. State what happens to chromatin so that individual chromosomes can be seen during mitosis. Chromatin coils or condense to become visible. It occurs during the prophase of mitosis. Figure 5.1 shows 2 human chromosomes at metaphase. You are asked to describe the structure of chromosomes at metaphase. Note that the question did not ask you to describe what happened in metaphase, but the structure of chromosomes. Each chromosome has 2 sister chromatids. They are 2 identical strands of DNA that make up a duplicated chromosome linked at the centromere. Each chromatid has a single DNA molecule. The DNA is associated with histone proteins. Histone proteins package DNA into structural units called nucleosomes, compacting it into chromosomes. Telomeres are found at the end of chromatids. They are the repeating non-coding sequence that act as protective caps, preventing DNA degradation during DNA replication. There is a mark for AVP. You can state that chromosomes are highly condensed at this stage. Question 6 is about HIV. The full term for HIV is human immunodeficiency virus. B. Suggests why treating people who have developed HIV/AIDS with ART may help to reduce the number of overall deaths from infectious diseases such as cholera, TB, and malaria. HIV weakens a person's immune system by reducing CD4 lymphocytes. When a person is immunocompromised, the risk of contracting other diseases increases. The impacts of these diseases would also be greater. HIV treatment helps maintain the number of CD4 cells, a type of T lymphocyte. This improves the strength of the immune system. The risk of having other opportunistic diseases will then be reduced. The chance of recovery is also greater. Opportunistic infections or diseases are infections that occur more often or are more severe in people with weakened immune system. There is a mark for AVP. For example, it can decrease viral load in the patient. HIV viral load is the amount of HIV present in a person's blood, measured by the number of viral RNA copies per milliliter. Figure 6.1 shows the coronary artery bypass graft. You are asked to show an area of the right coronary artery that has been bypassed. The coronary artery is a specialized blood vessel that wraps around the outside of the heart, delivering oxygen-rich blood to the heart muscle itself. I have highlighted them in red. You are looking at the heart from the front. Therefore, the right coronary artery is located on the left side of the diagram. The vein used to divert blood from the aorta to the artery is highlighted in blue. The yellow circle shows the region being bypassed, so placing an X anywhere near this region is correct. 2. Suggest and explain why it is important for the wall of the vein to become thicker after surgery. After being grafted, the vein is now playing the role of an artery. It has to thicken its wall to withstand higher blood pressure, making it less likely to burst. This will make the blood vessel last longer under greater pressure. There will be more smooth muscle and elastic fibers in the tunica media. The tunica media is the middle layer of a blood vessel's wall. Smooth muscle and elastic fiber can lead to vasoconstriction and elastic recoil. This contributes to faster blood flow. The changes are necessary because arteries shows a greater degree of vasoconstriction than veins. There will also be more collagen in the tunica externa. It is the outermost layer of the blood vessel wall. That's all for today. If you think my videos are useful, don't forget to like, comment, and subscribe. If you have any questions, feel free to ask me at the comment section. Thank you for watching and see you again soon.